Today I'm in Gangajal Ghati forest, which is situated in the south of West Bengal in Bankura district. It's predominantly a sal forest. You can see the density of the forest. And here I have come to collect my samples, leaf litter samples, which I had incubated 4 months back. And I normally come each month to collect one sample. I will show you how I keep my leaf litters in the forest for. The study is decomposition of the leaf litters to soil and the chemistry and the microbiology behind that. So he is Nimai Lak, my project assistant. He knows about the forest. He always assists me in the forest, and he is wearing the gloves to avoid contamination in the sample. Microorganisms from our hand should not enter into the sample, otherwise the study will be hampered. So this is the plastic within which we will keep the netted bags. What we do, we collect some leaves, a particular amount of leaf, and we put it into a netted bag, mosquito netted bag, and then we put it underneath the soil, 1 inch or 2 inch below the soil. And we try to make sure that animals and other intruders do not disturb the sample. So this is how we collect. This will be brought to the laboratory for chemical and microbial analysis. And now I will also show you. See, when you incubate your sample in the forest, you have to take care that you can relocate that place, because that is very difficult. In a dense forest, you most of the time you lose your samples. I have lost many such samples. So I decided to mark the trees like this and see, I will locate it for my next collection. So that's all about my forest collection process. Thank you so much. This is the bag.